All right, well, thank you for coming to the last lecture of CS 194. And thank you for your participation in this class throughout the semester. I realize Ben and I have been making up a lot of material as we go along, so sometimes it's been a little bit rocky. But anyway, hopefully you have learned something. It's my impression from the quizzes and homeworks and things that people have in general learned about parallel computing. I'm going to talk today. Oh, and we will be sending a link for the class review too. So you can all provide input on how we can make it better next time around. I'm going to talk today about how computing and data, and especially parallel computing, high performance computing, are used in COVID-19 research. This was a talk that I put together as a keynote back in about June. And things have changed since then. I've added a couple of things to it, but you can see as I go through this that there are a lot of places that parallel computing is being and has been used to address COVID-19. So one thing just to kind of let you know, if you don't already, that in addition to my role as a professor in EECS, I'm also the Associate Dean for Research in the Division of Computing, Data Science, and Society, which includes EECS. So EECS is both in the College of Engineering and in this new division, Data CDSS. And the iSchool is there, as well as statistics, and we're creating a new, something that's called the Data Science Commons, which will be for partnerships in projects such as COVID-19, climate change. That was one of the other talks that I said I could give. I've recently put that one together, and we'll be giving that in a couple of weeks, things like that. So we're really working to try to connect computing across the whole campus in social problems as well as scientific problems. So the other thing that I do is I work at Lawrence Berkeley National Lab. That's where one of my major research projects is. And I'm also a senior advisor on computing there. And one of the things that happened early on in COVID-19 is the national labs got together, the DOE national labs, that's Department of Energy, to figure out what we could do in general to help with the pandemic. And so what are the national labs? Well, this is just a picture that lists all 17 of them, or at least their logos. So Lawrence Berkeley National Lab is one of them, which is just up the hill. If you just go up Hearst Avenue until it kind of goes around the corner there, you'll run into Berkeley Lab. And that has about 4,000 people there that work on all kinds of basic science problems in physics and biology, chemistry, and so on. And that's similar to what many of the other multi-purpose labs do, but there's also some that are much more specialized in terms of focused on national security or focused on energy technologies and things like that. And just on the right are some examples of things that people in this lab, the national labs, have worked on. So all the labs got together and they created what they call a National Virtual Biotechnology Lab. So you got 17 labs spread out all over the country, and they weren't coordinated together. They all work on different projects. There's a lot of joint projects. But what came out of this was a set of projects that are spread out over multiple labs that were really organized nationally then to address things like medical therapeutics, so drug design for addressing the virus, manufacturing. This is actually a picture of test tubes, and some of the labs worked with industry, in particular Coca-Cola, which was seeing a drop in production of Coke and bottled Coke, and they didn't need as many bottles, but it turned out that they could repurpose the manufacturing plants to make test tubes, so those glass test tubes. And so some people at the labs worked with industry on that particular project. Epidemiology and testing, I'll talk a little bit more about that, as well as what is called viral transport, which is a group that there's a building up at Lawrence Berkeley Lab called the Flex Lab, which is designed as a very flexible facility for looking at energy efficiency, such as energy efficient windows and things like that. Well, they took that lab and repurposed it to look at how the virus flows around in a room to try to understand what risks are. And there are a number of other projects at other labs also looking at that, including computer simulations. So that's what the labs are doing as a group. What I'm going to talk about and just give you some examples of this work really fall into two major categories. The first one is at the biomedicine level. So things like looking at the viral genome, looking for drug design, looking for vaccines, And then the second group are at the epidemic level, so looking globally or nationally or statewide or countywide or whatever at how you manage resources, what are the interventions that a government can take in such as mask wearing policies or stay-at-home policies and things like that, and then trying to understand the economic impacts of the pandemic and how to address them in some cases. So as I go through this, I will try to also give you examples of what the computational problems look like and how they look like things that you've seen in this class. So this is, I think I showed this slide earlier. These are what we call the motifs of parallel computing. So dense and sparse linear algebra, which you know a lot about, especially the dense case, not so much the sparse case, but just think of that as, we did talk about that a little bit. Particle methods, which you looked at in homework 9 again, and also in one of the earlier labs. Structured and unstructured grids, so that's nearest neighbor sorts of things, which you saw on the quiz and elsewhere. And then Monte Carlo, which is a specific set of methods, but really we're going to think of it as all the pieces of these programs that are really embarrassingly parallel, so they have trivial sort of parallelization approaches to them. Spectral methods is one thing that we didn't talk about in this class. These are things like FFTs and cosine transforms, things like that. FFTs in particular, a big category of methods that are used, and those look, they have a pattern that looks a little bit like a sorting algorithm, so a radix sorting algorithm. So actually the butterfly, which I mentioned in one of the implementations of the barrier method. So it's got this butterfly pattern. So graphs and sorting and hashing, I'll say a little bit more about those. Okay, so one of the first things that DOE got involved with is a consortium that was actually led by somebody at IBM to put together a number of different companies, universities, national labs around the world, actually institutions around the world, to put together high performance computing capabilities, so machines and access to those machines that could be used for COVID-19 research. So there's 43 different institutions involved, and they have gathered together basically a commitment of computing time, which is equivalent to 7 million cores, 50,000 GPUs, and about a half an exaflop of computing and they have a central process then that people can put proposals into in order to get time on those, on all of those resources, which are all over the place. And there are 87 active projects right now. So a lot of different examples of things, and I will give a few examples from that. This first one actually preceded, I think, the formation of the HPC consortium and was done at Oak Ridge National Labs and by a group at Oak Ridge National Labs using their supercomputers. It's a molecular docking problem, and this continues to be an important problem that comes up in COVID and other disease research. It's looking at how molecules bind to various other molecules, and in particular the spike protein in SARS-CoV-2, which I'm sure you've heard about. And so you're looking for a molecule that's going to match with and then bind to that spike protein to try to prevent it from being contagious, to try to basically disable the virus. And what this team did was to screen 8,000 known drugs, so known compounds. And the reason that this is one of the major approaches that came up early on and comes up in any new virus or any new epidemic is to look for known drugs that can be repurposed, because then these are things that have already gone through certain levels of testing and are known to be safe, at least for certain types of uses, and it's a lot easier to get approval for them more quickly. They identified out of these 7,000, about 77 that were the most promising. This included some anti-aging drugs that I believe other people have continued to pursue as treatments for COVID-19. Now, it turns out that you can also use machine learning. So I didn't say what these molecular dynamics problems, these docking problems look like are particle simulations. And so when you've looked at these particle simulations, where you've got particles floating around. The exact rules on how far away you have to look at particle interactions, atomic interactions, will depend on exactly what you're simulating. But you've seen this pattern of parallelism. You divide up the particles, and hopefully you don't have to have every particle interact with every other particle, but can use some kind of a cutoff rule that will allow you to do something with a limited number of neighboring processors. Now you can also use machine learning, and one of the things that comes up in machine learning is you can look at these molecules as just a string of text, and you can think of this as you can do one-dimensional convolutional neural nets on them. So if you are familiar if you've taken 189 and you're familiar with machine learning, you may not have studied molecules, but you've probably studied images a lot. And even if you haven't, I'm sure you're familiar with image search. That's a nearest neighbor computation on an image. I've mentioned that a little bit before. You can think about these molecules as either being a one-dimensional string, this two-dimensional kind of image as it's kind of squashed onto the screen here, but they're really three-dimensional structures, and you can also do machine learning on these three-dimensional structures. And so there's a group that are using the HPC resources. These machine learning algorithms tend to be very compute intensive, and so they're looking at what are called distributed memory graph neural nets, which is something that one of my PhD students is working on right now for doing this kind of analysis of these graph structures. So think of a graph neural net as sort of like nearest neighbor computation, but instead of being on a two-dimensional image, it's going to be on a three-dimensional graph. And so you need to worry about sort of the connectivity of the graph. And maybe not surprisingly, it turns into a sparse matrix, and you've got a sparse matrix vector multiply, or actually sparse matrix times a tall, skinny, dense matrix. So that's what gets parallelized. And we have a recent paper actually that just got presented last month now, November, at the Supercomputing Conference on that. Now, one of my colleagues in EECS, Jennifer Listgarten, is looking at using machine learning to do design of drugs, both for COVID-19 and for other diseases. And what they're looking for is they're trying to solve what we're call an inverse problem, which is we know what we're trying to find something that will target the SARS virus. So maybe the spike protein in particular, although some people are looking at other parts of the virus that are haven't been as well studied, and you're searching through this space. So the previous work that I talked about was kind of a brute force approach. It says, let's just do simulations of all of these different known drugs and try to understand whether they're a good match for the coronavirus. And in this case, what you're doing is you're saying, well, let's look at the coronavirus and see what molecule could I design that would match that. And so you're searching through this enormous space of molecules. It's too expensive to do it exhaustively. So you use a machine learning approach to try to sort of navigate through the search space. And there's this problem, which is if you've got some data, you've got some known drugs, for example, and so if you stay near in the search space, near the things that you know, near the training data, then you have very high confidence that the machine learning method is going to give you good results, but you also need to explore other parts of the space. And so this statistical technique that Jen and her student Clara have developed is a way of, it's kind of a statistical spotlight, if you will. I also have a group that I'm working with along with some other senior researchers that are looking at these three-dimensional graph neural nets specifically for this protein structure, protein function determination problem. So how does this work? Well, we're also working on genome assembly, and I'll talk about that in a minute, at least analysis of the genome, and that's DNA, right? Once you've got DNA, if you probably had much better biology in high school than I did, because we know a lot more than we did when I took biology in high school, but you can then translate that piece of DNA that's in a gene. If it's a protein coding gene, you can translate it kind of in a pretty straightforward way into a protein. Now that gives you a string of amino acids. So you can think of it as just sort of like a string of text, which is great, except that we know that the function that a protein has is really determined by how it folds up and not just by its not just by the string nature. And so what this algorithm is doing is taking a graphical representation of that three-dimensional structure, as well as what you can think of as the text. So it's a kind of like a natural language model, and putting them together in order to automatically try to figure out, learn what the functional properties are of that protein. What does that protein do? Now, Yun Song, who is in the computational biology, the Center for Computational Biology, and also in computer science here at Berkeley, and sometimes teaches the machine learning and I guess maybe the algorithms course as well, in fact, he might be doing it this semester, is what looked at using machine learning techniques to try to improve testing. Now, they were specifically looking at serology tests. So these are the antibody tests. And it turns out that these tests look sort of like this. It's this stick. Each one of these is a different test. And it's got a score in it that shows up as a line that's sort of dark or light. And a human being, actually two human beings called readers, will read these things and say, that looks positive in these three different measures, C, G, and M. And then they take that data, and the way the tests had worked before is they actually just say anything that's positive based on what the readers tell them will be considered a positive test. And instead what Yun did was then they used some data because they had some people that were known to not have COVID because the blood samples were taken from last year. And then they had some known positive ones. And then they used that to train a machine learning algorithm that gave them much higher quality testing. There's also people using machine learning to automatically identify what looks like COVID-19 lesions in the lungs using CT scans, and also looking at trying to learn automatically which drugs are toxic using these graph neural nets that I mentioned before. Now, there's a great poster or kind of website that was put together by this group eLife, and this is just an image from it about the protein. It has a lot of other facts if you're interested in how big these things are, what their mutation rate is. This has been a big question, of course, from the beginning, is how quickly it mutates. So the problem is that if the virus mutates very rapidly, then you have trouble with a number of different things. One is you can have trouble with the vaccine. And it turns out if it mutates very rapidly, which fortunately it does not, then you would not be able to develop a vaccine because it was mutating more rapidly than you could make a new vaccine for the new virus. And it also affects testing and things like that. I'll say more about that in a minute. So here's some other facts, but I just wanted to kind of include this for reference. One of the things that people also did with molecular dynamics simulations then, so these look like these particle simulations where particles are all interacting with each other, the computing forces between these atoms, is to look at the dynamics of the spike protein and other parts of the virus. So how does that spike protein fold up and how does it change? So it turns out that it goes from a closed to an open state. And then the closed state, it's non-infectious, and then an open state, it binds to the ACE2 receptor on the human cells, which is what allows it to invade the cells. And so really these very detailed simulations. So you can see there's some fundamental science questions in here, as well as fairly applied questions that people are looking at. This was actually just came out a couple of weeks ago at the supercomputing conference that I mentioned before, where our graph neural net paper was, but this is the Gordon Bell Prize. And so every year at the supercomputing conference, one or more prizes are given out from Gordon Bell, who founded this prize. And it's for the largest or fastest computation done on one of these HPC systems for a real scientific problem. So unlike the top 500, which is looking at this dense LU factorization, so a benchmark problem, this is looking at new computational problems. And actually multiple finalists were working on SARS-CoV-2. And this one, the winner was this simulation of the spike protein and the viral envelope, which they were allowed to, they were able to see the sort of mucus around the envelope and things like that. This computation ran on Summit, which is the second fastest supercomputer on the top 500 list. So in the world, sort of the ones that end up on that list, at least, and that is at Oak Ridge, as I mentioned before. And it's a 305 million atom molecular dynamics simulation. Again, one of these sort of particle simulations. So fairly easy, as parallelism problems go, this is a fairly, these are fairly easy problems to parallelize. They're very compute intensive. They don't have very much communication relative to the computation, and that makes them scale very well on parallel computers. And here's another example, but this one is from Berkeley, sent from UC Berkeley from Ahmet Yildiz, who looked at modeling each step of the transition as it's invading a cell. So the binding to the receptor, to the ACE2 receptor, and then how it basically kind of opens the wall that cleaves the of the cell membrane, and then the membrane interactions with the protein, things like that. So there's a number of different proteins in the SARS-CoV-2 viral genome, or at least there are these protein coding genes, these genes in the genome that code for these different proteins. A lot of them have known structures that people have determined, say, through previous experiments. So that may be because the gene itself had appeared in other viral genomes, and over many years, people have done experiments. These are things like cryo-EM experiments, if you've heard of this. It's taking this electron microscope, freezing the sample, because a problem when you're looking at biology is if you shoot an electron beam at it, it tends to destroy the sample. So you're using these very cold samples, and then trying to get an image of it before it gets destroyed, things like that. And so this is some of the different structures that are in there. So as I said, the way that these things fold up are really important to what they do. And not all of it is well understood, but many of them are. Now, although it's not specific to SARS-CoV-2, I thought I would mention a major announcement that came out in the last couple of days. In fact, I think it was on Monday, shortly after it, or even during this lecture on Monday morning. DeepMind announced that they had won this contest to try to do protein folding prediction using machine learning algorithms. So this says now, if all I start with is a sequence of protein data, and I'm trying to figure out how is it going to fold, one of the things you can do is a molecular dynamics simulation, and you're looking at trying to figure out what is the minimum energy state of this thing and how it's going to fold. It's a very expensive computation, but more realistically, what people do to actually to get these protein structures is they'll use these cryo-EM systems and other kinds of instruments to try to actually take a picture of what the folded protein looks like. And this is instead saying, well, let me learn from the proteins that we know and their corresponding sequence and what they fold into. Can I train a neural net to predict what will happen with other proteins that I haven't seen before? And so this deep learning algorithm, AlphaFold, is outperformed 100 other teams by a couple of factors. So it was really significantly faster. There's a lot of press. All you have to do is type in the word DeepMind, and you'll see there's things in Science and in Nature and in a lot of the popular press as well about this announcement. A little concerned that it may be a little bit too much hype around it, but it certainly is a major step in understanding how proteins fold. And you can look at what I had mentioned before about trying to understand the function of the proteins. If we need the structure in order to understand the protein's functional behavior, it's very helpful if we can put this tool kind of in front of our machine learning algorithm in order to say, well, somebody else, this AlphaFold will tell us, given a string, a protein string, what it folds into. Now we can take that structure combined with the string itself and try to automatically determine function. Now, looking down kind of a level at the viral genome itself, there's a great website, and I highly recommend, if you haven't looked at it, to look at nextstrain.org. This is actually a movie, which I haven't captured the movie in here, but it shows on the left-hand side, this is the taxonomic tree of the viral genome and how it has mutated over time. So these are different branches of it as it's mutated. And then the colors actually correspond to the place where the continent in which that viral genome was measured. So this is taking things like nasal swabs from thousands, millions of patients, and sequencing the data. So the PCR test, which is used to determine whether you have an active case of SARS of COVID, and then take that viral genome and you map it back to a reference genome for that virus and try to figure out how it varies. So what are the variants? And those variants in the major variants will cause these branches in the tree. And so you can see how this map shows the different versions or variants of the viral genome correspond in the different parts of the tree here. So it's not perfect. I mean, the North America, the red you can see is spread out all over the place. That's because there were parts of infections that came from Asia as well as Europe and then across the US. But for example, the Asia one is pretty much clustered here at the bottom. So now we're doing string comparisons, string by string comparisons of DNA or of these, the viral DNA. And there's a project with a group that I work with at Los Alamos National Labs that has been involved. Well, I wasn't involved in this particular Edge Bioinformatics project. They took a set of tools that they had put together for other genomic analysis and made it available for hospitals and other labs around the country and around the world that were collecting this viral DNA, and they can upload it into the site, and there's various quality analysis, quality control that's done on it. One of the things you have to do is you filter the DNA to remove any human DNA. It's often contaminated both with the human DNA of the patient, which is self-identifying. So that's why it's thrown out so that you can then share the viral DNA without worrying about revealing the privacy of the patient, or the identity of the patient. But it's also contaminated with the DNA of the person collecting the data. So you can get human DNA mixed in a couple of different ways. And so this tool chain will filter all of that out and then put it together in a way that allows it to be, for example, aligned against the reference genome and used for things like that previous picture, although I think they're actually taking some of the data also from the GSAID data set. So at Lawrence Berkeley Lab, we had a project that was part of my Exabiome project that is using a tool called ArgWeaver to really try to understand the origins of the disease. So this is looking at the evolutionary history and where did it come from, things like that. And they have a new paper that is coming out on this as well. So this is now looking at things like a string to string, so DNA to DNA alignment. So from a computational standpoint, from a parallel computing standpoint, what does this look like? Well, you have on the left-hand side is a pairwise alignment problem. So let's say you've got two pieces of viral DNA, maybe one of them is the reference. So that's one that was collected early on and has been assembled, put together into a full reference genome by taking the pieces of the DNA and really fitting all the pieces together. So now you've got a complete reference genome and you've got a fragment of some DNA that has been collected from, say, a new patient, and you're trying to figure out, does it match? And to do this, you're allowed to have certain kinds of insertions and deletions and replacements in the two strings. So they're not going to be an exact match any more than our human DNA is an exact match for each other. But you run a string alignment algorithm, which is a dynamic programming algorithm called Smith-Waterman or some variation of that. So for example, anything along the diagonal here is that's a diagonal line is an exact match. The first C here matches the third C. This says, well, if I want to make this query match, I would have to delete the A and the B, then I would get the C, then I would add the B, then I would get the A, B, B, and then I have to delete another B, and so on. Not a great match. We would have much stronger strings of diagonal lines going in a real alignment if we thought it was a good alignment algorithm. And we have implemented this on GPUs. So Muaz Awan has done that on the NERST system, on Cori, as well as the Oak Ridge system on the Summit system, and using CUDA in this. And what he's actually doing is there's not enough work in a single alignment. So he's taking a bunch of pairs and aligning them in parallel on a GPU. So you can have a thread working on each one of those alignments. So it's a non-trivial problem. NVIDIA also has a library for doing that, but we have particular variations on the algorithm that he's implemented. And it also, by the way, can do protein alignment as well as DNA alignment. Now, if you've got a million of these things and you're trying to align them to each other. And now this comes up typically not in something like the SARS, the COVID use case where you've got a nasal swab and you're trying to match it against a reference of the coronavirus, genome. Then in that case, you sort of know what you're matching against. But in other cases, we're looking at environmental DNA and we're trying to figure out what does this match to? And we've got a huge database that maybe has a million different genomes in it. And we're trying to figure out what is this new genome we've just discovered. And so what is done there is to build a hash table. And that is storing fixed length strings that are called k-mers from both from the database and then from the thing that the query that we're looking at. And we use that to find something that has some common short string called a k-mer. In this case, in the picture here, I've just shown 3 mers. So you just say, well, if it turns out that it has AAC, then in the database, we would know that here's a genome that contains AAC. So we'll go ahead and run a more expensive alignment, which is an order n squared algorithm. So it's avoiding an order n squared algorithm to compare everything to everything, cuts that down and says, I'll only compare 2 things if they have a key in common. So the left-hand thing has been parallelized in a GPU. The right-hand, some parts of it have, we're still working on it. It's not trivial to try to parallelize hash table construction and lookup on a GPU. You know enough about GPU programming to imagine that that's a little bit tricky in a data parallel sort of style. Now, kind of moving up a little level from the viral genome, some people have used the HPC resources also to look at how these genes get expressed in the lungs of various patients. And this is looking at this thing you may have heard called the bradykinin storm, which is where your immune system, if you're a COVID patient, can kind of go haywire and really take over. And it's actually the immune system that causes lots of the problems. And what they have realized from these simulations, or were simulating, is that you get these leaky vessels in a COVID-19 patient that causes lots of problems, which is why you get multiple organ system failure in a COVID-19 patient. It's not just a disease of the lungs. And this simulation was also done by some people in my project, Steve Hoffmeyer, along with a group from the University of New Mexico and Arizona State. And this is you can think of it as sort of a particle simulation. So you're looking at the viral particles that are floating around through the lungs in this case. And then they interact with a particular part of the lung. But you're also then looking at all the different particles that are representing the parts of the various pieces of the immune system and what the immune system is doing in response to the invasion from the virus. And this was is a very expensive simulation. It was entirely written from scratch last spring well, actually, I think they're still working on various versions of it, and they're taking in image data from CT scans in order to get accurate models. They're basically doing what we consider validation of the simulation. So if you're trying to validate the simulation, you're trying to figure out, well, if you've got this simulation in which these particles are floating around and you think there are certain laws about what's going to cause them to move or cause them to attack or cause the immune system to be activated or whatever, then that will produce a certain, say, level of infection in the lungs. And then you compare that to what happens in an actual patient by looking at the images and see whether the simulation is producing the expected results. All right, so that's kind of biomedical level, genomics and the drug design and things like that. Not a lot of work that we've been doing on the vaccines. There's been so much work in industry on the vaccines that early on the national labs decided not to work on vaccines. I know there are people on the Berkeley campus that have been working on them though as well. So what about the epidemic level? One of the first things that I heard about from UC Berkeley here on campus was Bin Yu, who's a professor in both EECS and in the statistics department. And she and her group, a fairly large group of students and then collaborators from a number of different companies and other organizations, nonprofits and so on, were looking at hospital demand and the issue, which of course, unfortunately is coming up here again in the United States and was happening back in about March, was trying to figure out which hospitals were the most critically in need of face shields, masks, gloves, and other sorts of PPE, or also in need of respirators and things like that. So it turns out that this is a data problem. It's a big data problem, and there's a lot of data, but although the data is terrible, the data is really poor quality. A lot of the data, because there's privacy concerns about data that is collected by hospitals, it's very hard to get access to that data. There's data that's collected that you can see on websites. New York Times has a great data set, but it's very hard to get. That data tends to be about infection rates, which is highly problematic or noisy, because it depends so much on how many people are getting tested and how the tests are being used, who are they being given to, and their availability. And then death rates, which are also problematic, as I'll say in a minute. And also they're a trailing indicator, obviously. So by the time you know that the death rate is very high, it's too late to address the PPE. So they took a lot of different data sets from hospitals and counties, put it all together, did a lot of data curation. So this is really tedious, writing lots of scripts, I'm sure, and figuring out what data was good, what data was inconsistent, and what to do about that. They built a statistical model. Actually, that is they had 5 different statistical models, and they used a weighted sum of these, or a way of combining these different statistical models in order to predict the 7-day hospital demand. And they used that to then actually about 1 million face shields were flown around the world, based on some of the data that they had analyzed and the modeling that they did. Josh Blumenstock, who is in the iSchool, has done a lot of work, not just on COVID-19, but he kind of redirected some of his research into COVID-19 and works with governments in a number of countries that don't have very deep, large research programs, such as Uganda and Bangladesh, as well as Nigeria. And they're looking at, those governments were trying to hand out either microloans or very small amounts of money to try to help the poorest people in the countries. And they're trying to predict where are the most intense or largest populations of poor people in their country. They were looking at cell phone data. In this work, that they were also looking at satellite imagery data. So you can tell a wealthy region by how light it is at night, because if you've got money, you've got lights on, and if in the poor regions you do not. But you can also see the populous regions during the day. And so by sort of combining those 2 data sets, you can look at places where people really are densely populated but have very little money. So they use these techniques and have been using them to try to help governments identify places where there is high need. They've also looked at cell phone data. And in some of the countries, I think originally were just looking at things like very simple metrics, like what is the balance in your cell phone account, which of course might be a terrible indication of whether you actually have enough money. Although if you don't have any money in your bank, your cell phone account, that probably is a good indication of, that may be an indication that you don't have money. I guess if you do have money in it, that's an indication you're probably not impoverished, but it's very hard to tell from those alone. So they're combining these with other kinds of data in order to get much better information about things like where the call is being made and things like that, in order to get ideas of where people are clustered. So obviously huge economic impacts, and there are other people looking at this in both in the US and worldwide in terms of economic impacts of the interventions, as well as from the virus itself. Now, as I mentioned, the 3 metrics, that data kind of numbers that we often look at, are the death rate, the contagion rate, and hospitalization rate. And the hospitalization rate is a little problematic because the data often is not available publicly. The death rate is a trailing indicator. It also turns out that it's being underreported, and you probably have heard about this in the news as well. This was a study done early on by a computational astrophysicist who's in the physics department here, and but kind of redirected the statistical methods that they're using for things like looking at satellite image data. It used it to develop some techniques to correct the death rate. And they were looking at the data in Italy, and I know there's some other work on this now from Mike Jordan's group in computer science. And but in this particular study, what they were doing was correcting the data. And what they found, they were looking at what was the death rate a year ago. And it's more sophisticated than just sort of subtracting that off from the current death rate and then trying to figure out how many of them were caused by COVID-19, but looking at a couple of different statistical techniques for that. Not a huge computational problem in that case. And in another piece of work that is maybe surprising to me, even though I work on environmental biology, this is looking at the role of the background environmental microbe and how it impacts the severity with which people are getting or the contagion rate of COVID-19 in a particular region. So the hypothesis here, which I think even the developers behind this study, Ben Brown, who's in the statistics department here, as well as being at Lawrence Berkeley Lab and working with somebody at UCSF, was that, the question is, gee, is there any difference in how contagious these are based on what microbes that people have been exposed to in the past? And so they're looking at these environmental microbes and then trying to correlate that with how rapidly the disease is spreading. Of course, there's all these other factors they have to control for in terms of the spread, but they have found, it looks like a signal that says that there is a factor here in terms of the environmental microbiome. We do know, I mean, people have talked a lot about whether previous vaccines from other viruses, a flu vaccine, for example, or other things have led to differences in the contagion rate, but this is looking at the environmental microbiome. And the last thing I'll talk about here in terms of the big computational problems are simulations of the epidemic themselves. I may have used this quote here in this class before, but the quote is from George Box, a well-known statistician, who said all models are wrong, but some are useful. And it's very important to keep that in mind when we're looking at epidemiological modeling. So there's a few different approaches. There's what I'm calling here the curve fit or forecast, or the epidemiologists will often call a forecast. That's similar to, that's what Bin Yu's group was looking at that I told you about before. A number of other people, Maya Peterson and others in School of Public Health, have been doing those. Those are short term. They're sort of looking at a particular trend and they're looking at what direction things are going in, and therefore making a prediction of what things will look like 3 or 4 or 7 days out. ODE or agent-based simulations are what are called these SEIR models, where you have a population. So that in the agent-based simulations, you literally can have an agent, a little data structure that represents every person in the simulation. There's a very detailed one of these called City COVID, which is being done in Chicago. And it has a detailed map of what Chicago looks like, all the schools and churches and businesses and everything like that, and the houses. And then these things take in census data, so they know how many people are in the house and what their ages are. And they either have a more or less detailed model of where those people go to work. So you'll have a certain percentage of people that they're using transportation, mobility data, cell phone data to try to figure out where people are going to work or where they're going during the day. And then the model will have like a daytime and nighttime model that says, well, if you're in a household with somebody who is in the infected state, so a person will have a state variable that says, I'm susceptible, that means I haven't had it yet. I'm either I'm exposed, I'm infected, or I'm recovered. So that's the SEIR that's in here. And so then in each one of these states, if you're near somebody who is infected, then you can become exposed, and so on. And that's the way and you looked at this in one of the earlier homeworks I'm now remembering. So that's what those look like. One of the early models that was extremely controversial, this was one of the earliest kind of popular press releases, this was before the UK and the US really shut down a lot of businesses in, I guess, late February, early March. And this was a simulation from the Imperial College of London looking at what would happen with and without those interventions. So these models are very complicated, and they include things like household quarantining, social distancing. Some of them now have masks in them in terms of the models and things like that. And even this Imperial College one had, I know the time we were looking at it, had digital contact tracing that had been put in. And I'll say more about that in a minute. So I won't read through this, but it's about 14,000 lines of code. It's really mostly written in C, but it's kind of, well, kind of like what we've used in this class. It's a little bit of C++, but mostly C. And actually some of the code looks like it was written in Fortran and automatically translated. So a lot of array-based computations. And it's a fairly expensive simulation. They could run it on a large shared memory node for a US simulation. UK simulation was not a problem, but a full US simulation, we were looking at it for distributed memory parallelism. This is written in OpenMP, does not use GPUs, and does not use distributed memory. There's another simulation code, though. We determined it would have required a major rewrite to put it into distributed memory. This is kind of what I said before, except that there's the equations that tell you what's the probability somebody's going to get infected. For example, if they're in the same household with somebody who's infected, and so on. So you end up with this big sparse matrix, which is the infection matrix. And so if somebody is spatially nearby you, that'll be near the diagonal of the sparse matrix. And so those things will tend to be non-zeros or high values in that probability matrix. And then you're multiplying by that matrix in every iteration. And so one of the questions was, could we make that into a matrix that throws out the zeros that are, or the very small number of non-zeros that are farther away from the diagonal, because that gives you better parallelization. And this is just looking at what happens if you only keep 70% of the elements near the diagonal, or 90% of the elements, and how well you're doing relative to the full matrix. This was just sort of an experiment. And there was a team in the graduate class last spring that looked at this code and looked at both OpenMP analysis, and it turned out this little histogram on the bottom shows how effectively the CPUs are being used. You do not like to see these kinds of spikes with long regions in which very little is happening here. And this is the strong scaling plot here on the upper right, which shows that it's this is on looking at number of threads on a single core, on a single shared memory node, and we're up to 32, and we're getting up to about a factor of 4, is it? And that's because there's a lot of synchronization in the way this code is written, a lot of serial code that dominate. Amdahl's law is basically killing us in that code. And they looked at a number of different ways of trying to improve it using things like dynamic load balancing in OpenMP. There's a lot of features that are still missing. We make these models get improved all the time. The one that I'm working with, well, kind of loosely working with somebody else, Fred Streitz, who's from Lawrence Livermore National Labs, and he's got a team that's working on a code called EpiCast, and it's a distributed memory, so a high performance computing code. And they're looking at things like, people ask, what happens if we close bars? What happens if we close restaurants? Can you distinguish that from various other kinds of interventions? This graph, which is itself controversial, is looking at the viral load and contagion by age, or at least the viral load by age. And people have been trying to understand, and you see this in the headlines all over the place today, can we open schools for young kids? Can we open schools for middle-aged kids? What about college campuses? I'm sure something you all are aware of. Unfortunately, you're all adults from the contagion standpoint, and so it's much more likely that the younger kids in younger schools could probably open first. It's very hard to know, though, because they're so much more likely at a young age to be asymptomatic. So you get into all kinds of questions about the quality of the data again. But you can put this into one of these agent-based simulations. You can say, if somebody is in a certain age bracket, give them a different probability of being infected, and so on. I think I won't read through this. I want to just talk about some of the other things that involve computing. And before I do that, just mention that I think maybe I'll just end here, that these epidemic models are, these epidemiological modeling groups, there are hundreds of them, if not thousands of them at this point. They're not that hard to write. As I said, it was 14,000 lines of code, even in this very, very detailed one that had been written over many years. And similarly in EpiCast. So 10 to 20,000 lines of code is not very big for a scientific simulation. These climate models, and this is a picture of the International Panel on Climate Change, these will be hundreds of thousands of lines of code, much more complicated and built over a longer period of time. But I think the the reason I show this picture is because this is the size of the team, whereas most of the epidemiological modeling codes are built by a team of, say, 3 or 1 or 2 or 4 people, something like that. So really a small group of people. And instead of having, as we do in climate modeling, we have a small number of codes that are really national level or international codes that people in the science community agree on and have validated and can argue sort of reasonably from a scientific perspective about what are the advantages and disadvantages, as well as ensemble models that combine them together. In the epi modeling world, there's so many of these different codes that are not well vetted, and they're not being built on a coordinated basis. So I think we need a international kind of collaboration on this. And with that, I think I will end. I was going to say a little bit more about contact tracing and things, which you can now turn on your phones if you haven't. Happy to answer any questions about that if you have them. I looked into it quite a bit, and we tried to get a project going at Berkeley back in the spring, but eventually decided Google and Apple were going to go ahead and do this, which they have. But you can see there's a lot of different issues with the data problems and the computing problems in here, the large scale, the need to protect privacy, and something like the viral data, the need for, there's genomic analysis, there's molecular dynamic simulations, and everything else in between. So let me just see. I'm happy to see if there are any questions that anybody has. You can put them in chat, or you can just go ahead and unmute yourself if you want. All right, well, with that, we will continue to be in touch. Ben and I will be on Piazza. And I know there was a question about grading quiz 4. We will see if we can try to get that graded by tomorrow. It sounds like that will be a cutoff for you, but I need to talk with Ben about that to see whether or not that is practical. If you have particular concerns about grades in terms of thinking that you might want to switch to pass fail, you can also let us know in a private Piazza post, and we'll try to see if we can give you a little bit more feedback. In general, I expect the grades to be very high. I published a absolute grading scale at the very beginning of the semester, and just based on what we've been seeing the grades come in at, I think they will be very high, and we will that's fine. But so I don't think people should be too worried about it unless you feel like you have really missed a big chunk of the material and you're pretty sure you didn't do well on the quiz, or you are pretty sure you are missing a bunch of homeworks or things like that. So anyway, but feel free to reach out to us on Piazza in private. And with that, I'll just thank you all for coming and bearing with us during the semester. And I hope you learned some interesting things about parallel computing. Thanks a lot. Bye.